This podcast was produced by OAR FM Dunedin with support from New Zealand On Air.
Welcome to the OI Podcast, the Otago Institute podcast, in-depth conversations with experts from the worlds of arts, sciences and mataranga Māori, brought to you by the Otago Institute, the Otago branch of the Royal Society for Arts and Sciences. Kia ora, I'm Dr Barb, and I have got a quick reminder of our In Real Life seminar, complete with complimentary platters and a cash bar to celebrate a couple of amazing Otago researchers. It is the Barclay Theatre, Tuhura Otago Museum, 4 till 6 p.m. Wednesday, the 25th of June, 2025. We are so fortunate to have not one, but two of this year's Research Award winners as Professor Jemma Geoghegan, I got it, almost. The 2024 Prime Minister's Science Communication Prize winner, and Dr Olivia Harrison, the 2024 Prime Minister's McDermott Emerging Scientist Prize winner. And they are here to share insights about their research as they present Minds and Microbes, a multidisciplinary approach to understanding and managing anxiety and understanding the virome. Please book your tickets through the Tuhura Otago Museum website. It is free, brought to you in collaboration with the Royal Society Te Aparangi, Tuhura Otago Museum and the Otago Institute for Arts and Sciences. Last week we had a taste of this talk with Dr Olivia Harrison in the studio. If you missed that episode, it is always available via podcast. Don't worry, there are no spoilers. The talk will be completely different, focusing on that award-winning research. And today we have in the studio Professor Jemma Geoghegan. Oh my goodness. Welcome to the studio, Jemma. I am so sorry. I have just mispronounced your name so many times. And honestly, I've been practicing it for weeks now. I sourced. It's Irish, right?
It is. I'm
yes.
Scottish, really.
It's very confusing for me.
Yeah, just ignore most of the letters in there.
But yeah, Geoghegan.
Anyway,
Professor Geoghegan holds a BSc Honours in Genetics from Glasgow University of Strathclyde and a PhD from Otago University, which is awesome. And you've stayed here at Otago.
I came back.
You came back. All
yes.
right. Well, that's as good as staying here, isn't it?
Yes.
Welcome to the OI Podcast, Jemma. Hey, I have got a little bit of a blurb. Shall I read that first?
Sure,
so we've got kind of a...
Go for it.
Where we go. Isolated for over 84 million years, Aotearoa's unique mix of native and invasive species offers an exceptional opportunity to study viral evolution. By sampling viruses from a range of hosts, including birds, fish, reptiles and mammals, virologists like you investigate how viruses evolve, how host ecology influences viral dynamics, and what drives host jumping over time. Through your research, Jemma, you aim to uncover unknown viruses, identify potential disease threats, and understand the ecological and genetic forces which shape the virome. Wow. That is such a lot. So, yeah, Professor Jemma Geoghegan, the 2024 Prime Minister's Science Communication Prize for promoting public understanding of infectious diseases and advising a range of decision makers on how to respond to pandemic threats in New Zealand and throughout the Pacific. That is so much. I have so many questions.
That's good.
I just have so many questions, but I'm going to start right at the start with some basics. What is a virus?
Right. So a virus is a biological entity. It's not really alive because it needs to infect a cell in order to replicate, meaning make copies of itself and to sort of thrive and grow. So we call them biological entities, and they're not cells themselves. They need to infect a cell in order to live.
So what are they made of?
There's certain components of a virus that all viruses share. So they have a genome.
Right.
So like our genome is DNA. Viruses can either have a DNA genome or an RNA genome.
Which is sneaky, right? Yeah. Because we have DNA and RNA.
We do.
And I guess most living things do.
Yeah. And And all viruses, if they were DNA viruses like us, they would transcribe RNA as well. And some
We're not a virus, though.
we're not.
Oh,
We
sorry.
can be infected by viruses.
Viruses,
So viruses are the most abundant biological entity on Earth
Really?
because they can infect all cell types. So
Right.
there are 3 domains of life. So they
Right.
involve bacteria, archaea, which are a little bit like bacteria, and then eukaryotes, which means like multicellular organisms like
So
us.
the eukaryotes is basically everything else. Fungi, plants.
Everything. That's right.
Everything.
Everything. And viruses infect
And us, like
all
mammals.
of those domains. So everything we know about, viruses can infect.
Wow. It's like a whole separate kind of, like if you think of multi-universes, it's like a
Yes.
whole separate, like mirror world.
Yeah,
For
exactly.
everything there is, there's a virus that infects that thing. Is that right?
Yeah. Well, that's what we think.
That's what we think. I mean, why wouldn't there be if you can? It's crazy. Wow. Okay. So they're different from bacteria.
Yes, bacteria are cells themselves, and they don't need to infect a host in order to grow and multiply. Whereas viruses kind of need to infect a host cell. They overtake that host cell usually, and often they'll use components within that cell in order to replicate themselves, and then they'll burst out of that cell and go on and infect a new cell. And then when they do that within a host, then they often are transmitted to infect a new host and work in those cells.
Right. So, are all viruses bad?
No, hardly
Right.
any of them are bad.
Oh, really?
Yeah. So
Because I was just thinking about it, well, like bacteria, right?
Yes.
There's some good bacteria and some bad
That's
bacteria.
right.
And fungi, there's some bad fungi, like the ones that like infect your toenails.
Sure.
Or for a random example. And then there's, of course, the fungi that you eat and the ones
Yeah.
that you use to make
Yeah.
beer and bread and everything.
Yeah.
So we had quite a discussion about fungi with Professor David Olovich some podcasts ago.
Yeah, he's
and
great.
it was great because it was kind of the whole
Yeah.
life of it. And I was thinking, exactly, are viruses the same?
Yeah.
Are there good viruses
So
as well as bad viruses?
our knowledge of viruses is kind of hugely biased towards the ones that cause disease,
Right.
and the ones that are like economically important, so the ones that cause disease in us, like human health, or the ones that affect agricultural animals, things like that. And they're usually, we've discovered them because they're causing disease and they're causing a problem.
Right.
So our whole view of virology is skewed towards this disease-causing viruses.
Yeah.
And
And
in
not
actu-
just virology, right? But also like
yeah.
our public perception
Of
of,
course.
viruses.
Yes, of course.
I couldn't name you a virus that wasn't a disease.
Exactly. Right. So, but my research explores virus diversity that exists in nature. And the more we discover, that proportion of disease-causing viruses gets minuscule and tiny and tinier as much as we do this research.
So what do the other ones do?
That's a really good question. We don't really know. They don't seem to be causing disease in many of their hosts or most of their hosts. They seem to be a really important part of ecosystems.
Are they
And
sort of neutral? Like
They
they're
may
not
be.
doing anything beneficial, but they're not doing anything bad? Or do we just not know enough about them to know what they're doing?
kind
or?
of all of the above, really. So a lot of them are probably neutral. A lot of them might be even beneficial and providing some sort of function.
How could they be beneficial?
Well, we don't really understand the diversity of natural ecosystems.
Right.
We kind of see the things that we can see, but everything we can't see, we don't fully understand the
Right.
diversity.
So that's basically anything that we can't see as a microbe.
Yeah, that's
I
right.
mean, we can see it. It's just too small to see.
Sure. And so,
It's not like it's invisible. It's just too small.
yeah, exactly. They're real.
They're real. They're just too small for us. If we had like magnifying eyes,
Yeah.
or just a microscope,
Yeah.
or even something bigger than a microscope. Oh my goodness!
So, but we just don't know what they might be doing, because viruses have probably been around since the RNA world, like the first
Right.
things that
So
had
before the DNA
before
world.
the DNA world.
Right. So, because just let's go back to RNA and DNA and just do a refresher on how
Yes.
RNA and DNA work. Because RNA came first, right?
Yes,
We think.
we think.
We always premise everything with we think.
We think. Could be wrong. And so all RNA viruses share a common ancestor, or we think, to this RNA world. And so a lot of our genomes and the genomes of plants and animals that we live with have sort of genetic signatures of viruses from a really, really long time ago embedded in our genomes. And they're probably providing lots of important genetic functions that we just don't really understand what they do yet.
So there's bits of virus genome in our DNA.
That's right. They integrated themselves into our genomes and other genomes of plants and
How
animals.
do we know that? Like, how do you look at a genome? How do you see that's a bit of virus? Because it's not the whole thing.
Yeah. So, when we first started sequencing things, I say we, when scientists started sequencing things, like sequencing the Human Genome Project and all these things. Do you remember, people used to talk about junk DNA and the fact that, when they sequenced human genome and they found that, over 90% was this junk. Well, it turns out it's not actual junk.
See, that's classic science, eh?
We'll just leave
and then
it in the junk
we keep
box.
going and find out something more, and then that thing actually that we thought was really unimportant turns out that it's really doing something.
But so a lot of the time, this so-called junk actually shares genetic sequence similarity with viruses that are currently, not in the genome. And so, scientists around the world have been kind of tracking the insertion and evolution of these viral elements that have inserted themselves into host genomes over the years.
Right.
So it's become quite obvious that viruses, both the ones that are endogenous, they're the ones that have inserted themselves into genomes of hosts, and the ones that are exogenous, they're the ones that are replicating within our cells.
And getting that, so the exogenous ones have to get transferred somehow between hosts.
That's right. And so it's become obvious that they're hugely abundant, both of these types of viruses. And it's very likely they play important roles both within our genome and genome functions, as well as exogenous ones in ecosystems, and play some sort of role in the community diversity of that ecosystem. So we don't really know. And the more we discover, it's like opening up more cans of worms. There's probably a lot more questions than answers at this point.
Right. Okay, so it's really still in its infancy. And I guess that's got something to do with how hard it is to study them. Because if it's hard to see them, and then on top of that, it must be hard to keep them alive because you have to have them inside I mean, they're not alive to start with, but to keep them there so you can study them, you have to have the cells of the thing that they're...
Right. So until recently, until genomics technologies have come where they are today, it was really hard to discover new viruses because for the most part, you're right, the traditional way of discovering viruses meant that you had to isolate it from those tissues that it was infecting, those cells that it was infecting, and then try and grow it in the lab in cells that you had to kind of mimic what it was like for the host. And that's really, really hard to do. And so for the most part, you'd be unsuccessful. But then genomics technologies just revolutionized the study of virology in terms of...
So what is genomics technology?
Okay, the ability to generate genetic data and it can be from an individual or a population or an environment. So we use something called metagenomics. Now, so if you think of genetics being the study of genes and genomics being the study of genomes, metagenomics is the study of all the genomes within a sample.
Right.
And so if you go out and say take a nasal swab from somebody, that's how you test for things like COVID-19.
Yeah, so everyone has done that with COVID.
Yeah, right.
So that's like the classic, most fresh in our minds virus.
Right. So if you take a swab from somebody that's maybe got a respiratory infection, And you're not just looking for the virus that causes COVID-19. Maybe you're going to do metagenomics. That means you're going to sequence all the genomes in that sample.
Right.
And so what you would get from that sample is you would get a lot of the host genome. So if it was taken from you, you would...
So there'd be a lot of my
sequence
DNA in it?
Yeah, that's right. And then you would, if you had any bacteria up there or any fungi, you would get those genomes. And if you had any viruses, you'd get those genomes as well. Right. And so we're not targeting one type of genome. We're targeting the whole community of genomes in that sample. And so...
How many do you think you'd get just from a normal nasal swab? Like if I'm not actually sick?
Yeah, probably very few. If you're not symptomatically, showing anything. Because humans don't tend to carry a lot of viruses that don't cause disease. We carry some.
Right.
But not all the time, and usually our immune system kind of rids them from us.
And those endogenous ones that are kind of inserted into our DNA, when you do this sort of study, you just count them as part of
your DNA.
Us.
Yeah, that's
because it's already inserted.
Yeah,
so
it's not an infection anymore.
So in this study, you're just looking for those exogenous ones, right?
That's
so the ones that are kind of moving around from
right.
host to host.
So if I did that study and instead of taking a nasal swab from you, but I went out to a bat colony and like a roost of bats, for example, and I gathered up their guano or their poo and did metagenomics on that, I would expect to find lots and lots of viruses there.
Right.
Because bats, even though they're mammals like us, they seem to carry a myriad of viruses.
So just like a group of species that have more viruses.
Yeah, and there's lots of examples.
It's weird
like
though,
that.
Isn't it? Why would humans have less viruses than bats?
I think it's to do with our innate immune system. We tend to, rid ourselves of viruses that are causing any problems. We carry some viruses asymptomatically, but not
so it's not our opposable thumbs; it's our immune system that sets us apart.
Well,
we're not going that far. I can see by the look on your face you're not going to go that far.
Yeah. Well, there's lots of different host species that seem to carry lots of viruses, not just bats, but fish have a lot of viruses, birds, lots of wildlife.
Yeah. It's interesting. And maybe they've evolved to be able to just carry on their life with this viral load, whereas we're the opposite. Like, we just can't deal with it, so we just...
That's a potential hypothesis, yeah. I mean, viruses, their ability to become either more severely causing disease or not severely causing disease, this is the evolution of virulence, is really complex. And viruses are going to optimize their transmission abilities,
right?
No matter what. And if that
so they're not out to specifically get you as the host. In fact, sometimes killing the host is the worst strategy.
Yeah, so they're going to use a strategy that optimizes their ability to transmit. And so if that means killing the host, it increases their probability of being transmitted, that's probably what they're going to be selective for.
Right.
However, in a lot of cases, keeping the host perfectly healthy might be the optimal transmission.
Because then they move around and that's more chances that they'll interact with another individual of the same species.
That's
right. And so some are all viruses species specific, or are they generalists, or are they a mixture of both?
There is a mixture. So for a virus to infect us, it has to bind to a receptor on our cells to then enter our cells and replicate.
To the what of the cells?
A receptor. So it's basically kind of, if you think of it as a lock and key, right? So it has to have the right key to then unlock the right lock on the surface of our cells.
Right. And our receptors are different to the cell receptors of, like, my puppy, for instance.
For the most part. It depends on the virus. So I would say most viruses are very host-specific.
Right. Because it's quite a tricky thing to get into a cell, right?
It is. Yeah. You have to have the right binding capacity.
Right.
And so we are more likely to share viruses with other mammals, for example, because most mammals have quite similar cells, but not not exactly the same.
And of course, that's how they think of. Sorry, I'm interrupting there, but I'm just thinking that, like, as soon as you think of like sharing things, you think of avian bird flu.
Yes.
Right, that's a virus.
That's right.
And I think that's scary because lots of different birds have it.
Yes.
And there's a potential for it to jump to humans.
It is, yes. So ultimately, all viruses that infect humans have come from an animal at one stage or another. For a lot of them, we don't actually know where they came from, but they have all come from an animal.
How do we know that?
Because when we sample lots of animals, we see their evolutionary or genetic relations, or that their viruses, the genetic relations of viruses that we have are in other animals.
So don't we make any ourselves?
We don't make viruses.
Oh, we don't make. Where do they come from?
So we think RNA viruses in particular have all evolved from a common ancestor during the RNA world, which was a very, very long time ago.
Way back when?
Yes.
When there was just like
bits of nucleic acids.
of nucleic acids, and then there were viruses that use those bits.
Yes.
So before the cell evolved.
That's right. So we think that viruses, RNA viruses, share a common ancestor, and the way that they mutate and evolve allows them to diversify into different species and be able to infect new things.
Right.
And
are they, sorry, I'm just going to interrupt again.
Go.
Are they better at mutating than more complex things
Yes.
like people and plants?
That's
because
right.
they're smaller.
Yeah, there is this linear relationship between genome size and the speed at which they mutate.
Oh, right.
So most RNA viruses have very little ability for error reading. So when our DNA replicates and we make new cells,
Yeah.
if the DNA makes a mistake or a mutation,
Yeah.
for the most part, we have an ability to say, oh, it made a mistake, we're just going to kill that cell.
Right.
Because we don't want cells to be made with mistakes in them.
Right. Because most mistakes make you worse at being you.
Yes, that's how you lead to cancerous tumours and things like that,
Yeah.
right? So we don't want mistakes
Yeah.
in our genome, and so we get rid of them.
Right.
Viruses don't, or RNA viruses for the most part, have very little ability to do that. So they make lots of mistakes, but because they produce lots and lots and lots of progeny,
Right.
millions of progeny
So
in one
lots
go.
and lots of copies of the same virus as
That's
they're
right.
doing it.
But
Because
they
they're
make lots
small,
of
right? Because they don't
mistakes.
have to make the
Yes.
whole body
They're
of a
very-
person.
They're very small. So they can replicate really quickly, and they can kind of, the cost benefit is up in their favour because they make so many copies of themselves. But because they can make lots of mistakes, lots of those viruses, those baby viruses or virions, end up not being viable or not being selectively advantageous for that.
Right.
But then some do get that by chance, are selectively advantageous and allow that virus to explore a new niche or infect new cells quicker or infect a new host type, you know.
And if it's something that there's nothing there already, then it's just got that whole cell or host or whatever as a playground for itself.
Yes, exactly, yes.
So it just goes
Yes.
heaps and heaps and heaps of them.
So viruses can
Right, that
adapt
makes
really
sense.
quickly to different environments and to different host types. So they love to jump to new hosts because it gives them a whole new environment to explore, right? So viruses jump more than we thought they did.
Right.
Everything, the more we discover, the more viruses and hosts that we sample, the more host jumping we reveal. So viruses that have caused disease outbreaks in us, they've all come from an animal at one point or another.
And I guess the same with plants as well, like crop plants and things.
That's right.
So they're jumping this, like plant viruses are the same, are they?
Yeah,
as,
that's
as
right.
Animal
Yeah,
viruses.
yeah.
But plant viruses, because plants are so different from humans, don't tend to jump to
Yeah,
humans. They
that's
tend to
right.
jump to other plants.
That's right. So let's go back to the avian influenza virus or bird flu that you
Yeah.
mentioned. This is a bird virus. It transmits really readily between bird species,
Right.
and it is now spilled over to mammalian hosts multiple times.
Right.
most of the spillovers up until about a year ago were just dead-end spillovers. So they could spill over to the mammalian host, but the virus had little ability to transmit onwards between those mammals.
Right, so it got stuck at
That's
that.
right. So it was like a dead end.
So did it get stuck at that individual or that species?
So that's a good question, because about a year ago, these spillover events were happening to humans, and they were happening to, marine mammals a lot of the time. And then it jumped into cows, into dairy cattle, probably from infected poultry in farms.
Right, oh yeah, because they're quite close together.
That's
that
right.
makes sense.
And then cows developed the ability, or the virus in cows developed the ability to transmit between cows. And so for the first time ever, H5N1, which is the virus that is bird flu, was transmitting between mammals, and it's now endemically spreading in dairy cattle across the US.
So when you say endemically spreading, I always check this with microbiologists, because in ecology we talk about endemic species as species that, like an endemic New Zealand species, is a species that only occurs in New Zealand. But that's different when you're talking about endemically.
Yes. So I guess a virus, once it's endemic in a population, it is...
it sort of means the opposite. It means it's just there, doesn't it? Everywhere.
That's right.
It's very strange that you've taken that word.
This one we'll borrow from you.
Yeah, no. So right. So now so the avian bird flu is now endemic in cows,
in cattle and dairy cattle, and across the US. All states have reported infected cattle, and we think there's only been maybe 2 jumps from poultry into cattle. So,
but then it's spread between the cattle.
Yes, that's right, which is very worrying.
Because it means that it's evolved to be very good at jumping.
Between mammals too.
Between mammals, right?
So the good thing, I guess, is that cows have a really different respiratory system from us. And in cows, it's not a respiratory disease. So influenza causes respiratory disease among humans, right?
So that's the common flu, but also the Spanish influenza that killed thousands and thousands of people.
And there's been
swine flu.
There's been human flu pandemics every 20 years or so that we know of over the last century. But so with cows, it's infecting the udders, the cells in the udders, and it's about, in cows, there's about a 10% mortality rate, and it's spreading between dairy cattle by infecting, it's infecting the milk and all the machinery that goes along with milking cattle.
So once it's on a farm or a production unit, it's through the whole production unit quite quickly.
Yeah, and there's been now several reported cases in pigs. Now, pigs are the worrying thing because pigs have very similar respiratory tract and cells to us.
Yeah, they're always the thing that in medicine they use a pig to simulate a human before they...
surgery and everything. Yeah. But pigs are sort of thought of as this mixing vessel for new viral strains to emerge in humans. And this is what happened with swine flu in 2009. And pigs have probably been involved in every influenza pandemic in humans, but, our inability to sample at that time means that we don't really know, but we're just guessing. But once it's endemically spreading in pigs, that means it's probably not very far away from becoming a human pandemic. And
so how worried should we be about that?
I'm quite worried. I think that it will become a human pandemic. You probably don't know.
I mean, a 10% mortality in cows...
It's quite high, isn't it?
That's quite high. That's 1 in 10.
Yeah.
That seems like...
Yeah. So with bird flu, we've known about highly pathogenic avian influenza virus or bird flu since about the 90s. And since about 2002, we've had quite frequent human cases, all linked to infected poultry.
Right. So poultry farmers.
Yeah, that's right. So people with direct exposure to infected animals. And it has had a really high mortality rate in the people we've known to be infected, about 50%.
Wow. So that's even worse.
Yeah. But
of course, the one that comes through cows or pigs will be slightly different. That's that whole variant thing, isn't it?
That's right. And it's usually been kind of spillovers that are relatively contained to poultry farms.
Right.
But what's happened with this latest bird flu, and the reason why everyone's talking about it now, is because a couple of years ago, actually around 2020, when the world was focused on a new coronavirus.
Yeah. COVID.
At the same time, a new lineage of bird flu emerged, and it's first spilled over from wild birds. So wild birds have avian influenza virus usually, and they don't get sick at all from it.
Oh, right.
And it's
because I know there's a bit of worry in the conservation
huge worry,
world in New Zealand, and we'll get onto that a bit later. I'll let you finish your thought. But
so what happened around 2020 is there was a spillover again from wild birds into poultry. It becomes really pathogenic in poultry. Some of these H5 and H7 are the two main subtypes that become really pathogenic. And so it killed a bunch of poultry. But what happened, we think, is that once it became pathogenic, it actually spilled back into wildlife. And then it became really highly pathogenic in wildlife for the first time.
Okay, so it was quite benign to start with, and then it sort of flipped out and then flipped back in.
That's right. And so this lineage has been spreading around the world for the past few years, and it's been killing millions of birds, wiping out entire populations.
Right.
So it's now spread to every continent apart from Oceania, even Antarctica.
Which is us. Oh, my gosh.
That's right. So it's now in Antarctica spreading. So
really? So it's in the penguins in Antarctica?
Yeah, and a few other different
the skuas
species.
And things.
That's right. And it's killed thousands of marine mammals, like fur seals and things like that in South America.
That's crazy, because you really don't hear that much about it.
Yeah, you don't, do you?
Yeah, because I just know because of the conservation world that there's quite a lot of kind of worry about it in New Zealand, because of course a lot of our bird populations are endemic to New Zealand, so they're only found here.
That's right.
In that other use of endemic in a confusing broadcast. But also quite a lot of our bird species have really small populations and quite stressed populations through other things like predators and habitat loss.
Over 80% of birds in New Zealand are already on the brink of extinction.
Yeah, so that's that doesn't count all of the ones that have already gone extinct as well. So
a new virus where our birds have never been exposed to it before has a potential to be catastrophic, I think. So we don't know how it will get here. At the moment, it remains very much a bird virus. And so a lot of our research looks at migratory birds, sampling migratory sea and shore birds and also sedentary birds here to see what types of viruses they have and understand the transmission, they're globally connected viruses, to see how it might come here, what sort of species might bring it to New Zealand, and so on. It could also come through migratory or marine mammals that disperse into New Zealand and the subantarctics and
then go back to the birds, because of course I guess there's a lot of those marine mammals and birds live quite close contact.
Yeah, and lots of birds have been infected. Lots of marine mammals have been infected by feeding on infected birds.
Those omnivore / predator
and
mammals.
Then so that's how it'll probably come here, but we don't really know the ability for migratory bird, for example, coming from the Northern Hemisphere might take a couple of weeks to get here.
Right.
Before, this lineage emerged, it was thought that it would, if it was infected by a highly pathogenic bird flu, it would die on the way here.
Right. But so that might be why we've lasted so long without it.
That is why, because we don't also have any migratory waterfowl, the ducks and swans and things like that most countries have. And those are the types of birds that are the natural reservoir host for this virus.
All right. So that's quite lucky. That's just
so we are quite geographically isolated, but we do have lots of migratory birds that connect us to the world. And with this new lineage, it continues to surprise us in ways that birds could remain asymptomatic with the virus, even if it's highly pathogenic. And
we can't put them in isolation. We can't lock them up in a hotel for 2 weeks.
Yeah, exactly.
What can we do about it?
I think can
we make vaccines?
Yep, there are actually vaccines already, but we can't use them. Well, so it's complicated. So there are some vaccines, and
because it's not like COVID that wasn't
Yeah,
than it
it's
was.
not a new virus.
It's a
We've
virus
known
that's
about
everywhere
this.
else, so
Yeah.
it's quite well studied,
That's
I imagine, the
and
virus.
we've had, seasonal flu vaccines for ages, right?
Right.
You should all go and get one.
Yes, I've had mine.
Yeah, great. And so,
I like my seasonal flu vaccine because I really don't like being sick.
yes, the flu really sucks.
Yes.
So basically, with the vaccinating wildlife, it's tricky, obviously, but logistically, but also it's more complicated than just vaccinating everything. So at the moment, DOC are doing a trial of using a vaccine for 5 different endemic species in New Zealand, but only for 5 individuals of each of those species, and only individuals that are completely non-wild, so in aviaries and sanctuaries.
So ones that, for whatever reason, are already removed from the population.
That's right.
So either they've been raised in captivity because they were sick when they were babies, or
that's right. So they're
something along
not
those
wild
lines.
animals, but they are being. So they're trialing a few vaccines.
Because do you need a different vaccine for each species?
No, they've used. They've just used avian.
The same vaccine.
Yeah, that's right.
This is a thing that I would like to clear up. Just while we're talking about vaccines.
Yes.
So there's 2 different types of vaccines, right?
That's right.
There might even be more.
Yeah.
But there's what they call live vaccines.
Yes.
And what they call not live
That's
vaccines.
right. Yes.
Because the other thing I've just had is a shingles vaccine.
Oh, that's good.
Because I was unaware of how easy it is, like what your probability of getting shingles is.
Yeah, once you've had chickenpox.
Yeah, so if you've had chickenpox, it's the same virus.
Yes.
And then it pops up. Like you get chickenpox usually when you're a kid, if you get it. And then shingles when you start to get older.
Yes. Or it doesn't have to be age. It can be if you're immune compromised in any sort of way, or stressed, or
overstressed
going
or
through
something. Yeah. It's interesting,
Yes.
But yeah, it's for my age bracket, it was 1 in 3 chance of getting shingles.
Yeah, it's really
and it
high.
can be really nasty.
Oh, it's awful. And it lasts for a long time.
And it lasts for a long time.
Yeah.
And it's kind of that virus, just that chickenpox virus, just stays in you.
Yeah.
God, it's
Pox
horrendous.
viruses.
Anyway, the lady said that was a live virus.
Yeah, live attenuated vaccine.
Yeah. So
So
explain. Because you're not get like that, like it's that attenuated, which is the important bit, right?
Yeah, so you're not.
So
you're giving
it's
someone
attenuated
enough-
just means weakened, right?
Yeah, so it's not. It doesn't have the ability to cause an infection. It just sparks enough of a reaction from your immune system to basically build up some immunity. So that if you were,
it's like a kind of warning shot from the far off hill, right? So it's like, hoo-hoo, I'm here, and you're like, you shouldn't really have told me because now I'm getting myself all ready for you, right?
Yeah. But so that's the way live attenuated vaccines work at very brief, high level. But for most part, lots of people in New Zealand got an mRNA vaccine for COVID-19, for example.
Right.
And an mRNA vaccine is very different. It's you're giving with COVID, or the virus is called SARS coronavirus. You're giving someone the RNA or the messenger RNA to make a spike for binding to our cells. So you know how I said that you had to bind, a virus has to bind to our cells. They have to have.
So the cell, the human cell,
yes,
has like a coating around the outside of it, which has spikes on it.
No. So the virus has spikes on it.
The virus has spikes.
So let's just say that's the key. Okay.
Yep.
And our cells have a receptor. Say that's the lock. Okay. And the virus that causes COVID-19 has all these spikes, and they have to bind to our receptor to be able to enter it. Right. So our receptors are called the ACE2 receptor for COVID-19.
Right.
And
why would we have a receptor for a virus?
No, you're asking the wrong question. It's how clever is that virus to evolve that spike to bind to our receptor?
Right. So the receptor was already there, right? It was there for another reason.
Yeah.
And the virus is just
clever.
So it's not really clever because it's not alive. It's just the ones that were able to do it successfully have managed to.
That's right.
replicate themselves a
That's
lot.
right.
And they have
beneficial for them.
Yeah, because once they can do it, then they can get into that cell, right?
And lots of mammals have ACE2 receptors.
Right.
That's the name of the receptor that we have. So, people think that a lot of people have speculated that this virus was grown in a lab and so on. But, lots of animals have these receptors. When it first emerged into humans, it wasn't it wasn't designed for humans, right? I mean, over the past 5 years of this pandemic, we've been tracking the evolution of this virus, and it looks a lot different now. It's, acutely tuned to infect human cells. When it
first came out, it wasn't.
No, not at all. I mean, it was pretty good.
It was pretty good because obviously it was actually infecting humans, right? But it was bad enough at doing it that it kind of was a little bit clumsy. It looked more like it
Sure, that's right.
So can you use that kind of studying of the genome of it to figure out what species it came from?
Yeah, well...
or most likely came from.
Yeah, but there's, after the pandemic emerged, there was sort of mass virological sampling of lots of different wildlife, from bats to, animals that were traded in live animal markets, lots of wildlife sampling to try and understand the closest genetic relatives of the virus that causes COVID-19 in humans. And the closest ones are viruses that have been sampled in things that were traded in live animal markets.
Right.
Things like ferret badgers and raccoon dogs and things like that. They're being
what
traded.
What do you use ferret badgers or raccoon dogs
I believe for the fur.
Oh, right, okay.
I think. But I think also
is it a
bushmeat is also...
Do you mean ferret and badgers as two separate things, or a ferret badger?
There is such thing as a ferret badger.
Wow. And a raccoon dog is like a dog. I don't know. It's crazy. This is why I went into botany, right? Okay. So anyway, there's like all these live markets basically with quite stressed out animals because they're in close proximity to other animals.
When humans come into contact with wildlife, we generally haven't lived among lots of wildlife.
Right.
And there's only certain communities that have. And so for the most part, the viruses that they have are very different from the viruses that we're usually exposed to.
Right.
But usually, there's a lot of mammalian wildlife. So our host cell receptors are quite similar, for example.
Right.
And so when we do come into contact with them, whether it's through animal trading or encroachment on their habitat.
Right. Or like you were saying before with the poultry farmers and the avian flu.
That's right. So when we're, come into contact with animals, we're increasing the risk of transmitting viruses either from them to us or us to them.
Yeah.
And so...
But there's more of them and they have more viruses, so it's more likely them to us. And plus we notice it when it comes to us.
Yes, we notice it.
Right.
So, and it doesn't have to be wildlife. It can be farmed animals as well. Like livestock give us lots of viruses and bacterial species as well. So, yeah. So I was talking about
mRNA. And what is mRNA and why is that different from a live attenuated virus?
Yeah.
That's where we sort of diverged.
Yes, massive divergent.
So the M in mRNA is messenger RNA.
Yeah.
And that's just in your cells normally.
Yes.
That's the stuff that's kind of going to our DNA and saying, hey, this is a copy, make new ones somehow.
Yeah, something like that. Yeah.
You explain it better, Jemma.
No, you kind of got it right. So it's basically telling the DNA to transcribe into RNA to then the RNA then, makes a protein and that protein performs a function, like if it's an enzyme or whatever. So it's basically the way that our DNA is used for functions in our body. And how... the virus is an RNA virus and it has things all around its surface called spikes. Right. And these spikes are the things that bind to our cells to infect our cells. This is just for coronaviruses, by the way. And so what the mRNA vaccine does is it injects us with some messenger RNA, but only for the spike. So not for the rest of the
Not
virus.
for the rest of the virus,
No.
just for the spike.
Just a tiny bit.
So the spike itself doesn't do anything
No,
to you.
not a
It doesn't have the replicating
it doesn't have
instructions.
any. Yeah, it's
it
just for the
doesn't have
spike.
the make you sick instructions. It just has
That's
the
right.
instructions for this tiny little spike.
That's right.
Which is the key that matches to the lock.
Exactly.
That's already on our cells
Exactly.
for a totally other reason.
Yes. So then when it makes some spikes, and
Right.
our cells have the ability to recognise that spike. But we
Right.
don't get
So
sick
it's
from
it's just the spike that's floating around, right?
That's
so this mRNA is saying to our proteins, to our body rather, make these proteins in the shape of a spike.
No. So the vaccine it basically makes spikes, and our cells have the ability to recognise the spikes.
Right.
And it kind of gives us an immune response. So
Right.
that
So our immune system is going, hey, there's this foreign thing.
Yeah.
It's a spike and it looks like this.
Yeah.
And then it sends that round to all the immune cells and says, hey guys, just keep this in your back pocket, because if you see one of
Exactly.
these, it's not part of us.
Yeah.
It's a foreign thing, attack it.
That's right. So when we do get infected with the virus, we'll be like, oh, that's that spike again. Let's make sure that doesn't get
Right.
infected.
But we've already got the proper weapons set up.
That's right. Yeah. So that's the kind of difference between
So
those
why do we need to
vaccines.
keep getting new COVID vaccines?
Yeah, well, remember I said that, RNA viruses in particular don't really have an ability to correct themselves when they make mutations or mistakes in their genome. Well, that means that they evolve very quickly. And so
Right.
the the vaccine that you had, a year ago, or the infection that you had 6 months ago is from a virus that's quite different from the variants that are circulating today.
Right, because they've changed.
Yeah, that's right. So they've changed so much that...
So what are they changing? They're changing how their spike looks?
Well, they're changing quite a lot of parts about them, not just the spike, because that's how they enter our cells, but also other parts. So the reason why they have to keep changing is because there's this sort of evolutionary arms race, because we're constantly subjected to, infection or vaccination, and we're exposed to the virus. And so our immune system gets really good at recognizing the virus,
Right.
either through past infections or vaccinations.
And
So-
as we get better, then the one that was there is worse
at infecting us.
at infecting us and replicating itself.
That's right.
And so some little mutant from one of those other little things that had an error in it
Yes,
suddenly
is better.
is better
Right.
because it's just enough different that our immune
Yeah.
systems can't recognise
So it's
it.
constantly trying to find ways of evading our immunity, and we're constantly trying to find ways of blocking infection.
Right.
And so we're constantly keep changing, and
Does that mean we'll have to have COVID vaccinations forever?
unless it's somehow eliminated or comes
maybe-
to a point of not causing severe disease.
Yeah, I was going to say maybe it'll
potentially,
just
but
because it doesn't actually benefit from us being sick, right? That's just
again
a side effect, right?
It's
If it could pass itself on better without making us sick, it would do that. It would
Yes.
evolve to do that.
But, there is this sort of long-standing fallacy in virology that viruses always become attenuated over time, which means they always become less severe over time.
Right.
But that is false, because the selection forces on viruses are transmissibility, right? And so
Right.
whatever allows that virus to transmit more and to
Right.
infect new cells more, that's what's going to happen. And
Right.
with COVID-19, it's quite interesting because for the most variants, you're more contagious before you even have symptoms. And so
right. Because that
most
makes
people-
sense, because if you isolate yourself after you've had symptoms.
Yeah, I mean, most that was quite surprising, I think, for this field. Most viruses, you seem to be quite symptomatic when you're most infectious. But for the most part, this virus was studied a lot. I mean, basically everybody stopped what they were doing on everything else. That's right.
And because a lot of people say, oh, how did they possibly manage to get the virus, the the vaccine rather, for this virus so quickly. And it's basically they stopped doing everything else, threw all their people and all their money at this.
I mean, it's interesting because people had been working on mRNA vaccines for ages. I mean, a lot of the work was done on Ebola virus, and there was, getting to a point of rolling out potential vaccines for that, the mRNA and things like that. So our knowledge of this virus didn't just
start when we first heard about COVID, right?
You know, everything can be applicable from the virus you were studying before COVID, change it to this virus, and that's why it was so advanced when we were,
And mRNA, like the whole idea of mRNA vaccines itself, that wasn't a new thing at all. That was well studied.
Exactly. So anyway, can't really remember what we're talking about.
Sorry, Jemma, I've interrupted you. I'm terrible. Oh, my goodness. If only we could play this back and see where we were. Anyway, hey, we're coming to the end of the show. Unbelievably. It's got totally sidetracked because, of course, you are a bit of an expert on COVID. And it is great to take advantage of that and just clear up some of the things. You were talking about how it's a fallacy that all viruses become attenuated or less severe with time.
Yeah, that's right. So, we've seen this in the early sort of years of COVID, where the first variant, we call it sort of Wuhan variant when it first emerged. And then we had a few different Greek letters. We had Alpha, which seemed to be a bit less severe than the first one. And then we had Delta, and Delta was more severe because it infected more deep lung tissue and people were getting really sick. And then we had Omicron, which was really good at infecting our upper respiratory tract cells. So it was less severe than Delta. So we've seen this kind of different...
Up and down.
Yeah, and different levels of severity. But because of COVID, most people are more contagious before they even know themselves got sick. So they've done lots and lots of their transmitting before they get sick.
Which is, of course, why we had the lockdown, because if you don't know you've got it, you can't know you're transmitting it.
Yes, that's right. So, lockdown's very effective, it turns out, at stopping, eliminating the virus from the country. And so we... there's no sort of selective pressure for the virus to keep the host alive when it's already been transmitted.
Right.
So it doesn't really matter if it.
So it's not really that the virus benefits from you dying or being sick, or not. It's not
in this case. Yes,
it's just kind of neutral to it.
Yeah, I mean, I guess it's really good strategy that it keeps you completely asymptomatic while it's doing most of its transmitting.
Yeah, that is a really good strategy. Yeah. We should be quiet about it so the other viruses don't hear too much about it. Yeah, I think that in some way the lockdown was so effective here in New Zealand that in stopping the worst of the virus, because of course we've got COVID here, and lots of people say, I've got COVID now, I've had COVID, it wasn't worse than the normal flu.
That's because, so in the first wave of COVID, think back 5 years ago, right? Yeah. And the whole country had to lock down and we were able to eliminate the virus. But, then no one had any exposure to this virus at all. Until we kind of opened up and turned away from this elimination strategy to more of a mitigation strategy, we had 97% of vaccination coverage in New Zealand. So when, until then, we only had a couple of thousand infections. And after that, that's when everyone got it.
Right.
But most people had already had a vaccine.
Right.
And so you experienced your first COVID infection after you'd had vaccine, right?
Whereas, so the amount of badness that it was to us was much reduced.
Totally.
And still some people got really sick.
Oh, yeah. I mean...
But it's all a numbers game at that point. It's not about your individual, how sick you felt. So there may have been some people who weren't vaccinated and also didn't get it too bad.
That's right.
But that's just a numbers game, right?
That's right. I mean, you look all around the world in early 2020 and people, there was mass graves and people were dying in hospital corridors because this virus was sweeping around the world, with a very high mortality rate because no one had any exposure to this virus or a vaccine before. So we experience a very different pandemic, and it's easy to forget that.
I think so.
Yes.
Hey, I'm going to have to unfortunately end on that note. Thank you very much for coming along. You've been listening to the OI podcast. If you enjoyed this episode with Professor Jemma Geoghegan, we think you'll enjoy some of our other Royal Society Research Award interviews. Last week's episode, Anxiety with Olivia Harrison, Speaking for the Trees with Dr Peter Heenan, and International Relations and Public Diplomacy with Professor Natalia Chaban, or A Genetic Cause for Stomach Cancer with Perry Guilford. Don't forget to book tickets for our In Real Life seminar, the Barclay Theatre, Tahuro Otago Museum, 4 till 6 p.m, Wednesday the 25th of June. And remember, you can support the production of the OI podcast by becoming a member. Just pop along to our website, Otago Institute. Thank you very much. Hey, ka kite.
Bye.
You've been listening to the OI podcast, the shout out for the arts, sciences and Mataranga Māori.
You can support this podcast by becoming a member of the OI. Check out the Otago Institute for Arts and Sciences on Facebook or our website.
If you'd like to listen again to this episode or others from the series, look for the OI podcast on OAR FM's website, oar.org.nz.
Brought to you by the Otago Institute for Arts and Sciences, the Otago branch of the Royal Society of New Zealand.
This podcast was produced by OAR FM Dunedin with support from New Zealand On the Air.